So this video is going to explain mutations and basically the result of mutations. So if you think back to what a mutation is, it's a change in the sequence of DNA. So let's go through what some possible consequences are if we actually change the sequence of the DNA. So I have two situations for you to take a look at. In situation one, it says, what would be the result if the first C in the mRNA sequence was read as a G? So what that means is this very first C here, we're not going to read it as a C, but it's going to be read as a G. So maybe that means DNA polymerase didn't catch that mistake, the wrong base was accidentally brought in, or there's a mistake in the anticodon coming in. But if you take a look at situation 2, it says, what would be the result if we take that same first C in the mRNA sequence, but we delete it? So all of a sudden that C is completely gone. So I'm going to give you a couple minutes to go through and actually figure out what the amino acids sequence would be. So split them up into the codons, and then figure out what would those codons code for in terms of the amino acids. So I'll give you a couple minutes, so hit pause, use your codon chart, and then figure that out. Okay, are you ready? I'm about to show you the answers, so let's see if you guys correctly figured it out. So let's take a look at situation 1. So if we change the C to a G, what that means is this second codon is going to end up coding for a completely different amino acid. Now, depending on the protein, depending on the amino acid, sometimes making that slight change doesn't completely throw off the protein. However, sometimes changing it by just one base and just one amino acid will completely shut down the protein. It completely depends on the protein. Now, one thing to point out is sometimes if I change one base, I actually get the exact same amino acid. Because remember, there are some duplicates in the codon chart. So there are some mutations that literally have no effect on the organism. Now, there are some mutations that just slightly maybe change the color a little bit or the texture, and that's what creates things called variations. And variations are actually very important for the survival and evolution of a population. So let's take a look at situation 2. So remember, we just completely got rid of this cytosine. So now what ends up happening is all of my codons shift on over. So what ends up happening is now, yes, this codon is changed and we get glycine, and then this codon should have been valine, but now we get a stop. And then this one's changed, that's valine, and then that's not even a codon anymore. And so because the stop proton came a lot sooner, and basically every single one of these codons is changed, I have tons of wrong amino acids that are being brought in. So let's take a look at question 3. It says, which of these two situations do you think would have a more serious effect on the resulting protein and why? Take a couple seconds with your partner and see if you can figure that out. Okay, you ready for the answer? I would definitely say situation number 2. Okay, situation number 2 is called a frame shift mutation because basically the whole thing was shifted on over, which means that every single codon ends up getting changed and all the amino acids getting changed. This usually causes that protein not to function at all. So that's a really serious effect. Point mutations, which was like in situation 1, is basically have one point where there's an issue, where the rest of the stuff is fine, but I've got one point where it's messed up. Most often, these point mutations are not as serious. So that concludes the video on mutations. Take a look at the SNORC activity inside your Schoology folder, because that's what you'll be working on next.